At first, I'd like to thank Sophion Bioscience to organize this conference and inviting me here. I'm very honored to present here. Today, I'd like to talk about TRIM2, one of TRIP channel having a temperature sensitivity to body temperature sensor range. TRIP channels have 6 putative transmembrane domains and 4 domains between the segments 5 and 6. 4 subunits are considered together to function as to form a functional channel. Many of TRIP channels are non-selective cation channel having the relatively high calcium permeability. And so their activation causes the membrane depolarization and intercellular calcium elevation. Currently, 11 members of TRIP channels are known to have a temperature sensitivity and called sum-TRIPs. Each sum-TRIPs have a characteristic temperature threshold. In the case of TRIP V1, the current is activated when temperature is increased to 43 degrees Celsius, like this. And this traces indicates the temperature activated current of some TRIPs accompanying temperature increase or decrease. And friction point of each channel indicates the some temperature threshold for each channel. And many of these some TRIPs is expressed in sensory neurons and is involved in sensory somatic some sensation. In addition to sensory neurons, skin keratinocytes are also considered to receive the temperature change and transmit the information to sensory neurons. Employing a series of some TRIPs makes it possible to detect and discriminate this wide range of temperature. Interestingly, many of these some TRIPs are also expressed in cells and tissues, which is kept at the core body temperature like this. One of the aims of this, our research is to reveal the mechanism regulating activities of some TRIPs at core body temperature. Today, I will talk about our research focusing on TRIM2, a body temperature sensor. TRIM2 is one of some TRIPs as a non-selective cation channel. The activity of some TRIP channel is regulated by many endogenous factors, including cytosolic ADP levels, calcium ion, and reactive oxygen species together with temperature. Because TRIM2 expressed in cells and tissues, which are kept at core body temperature like this, the activity at the body activity of TRIM2 under the body temperature should be precisely regulated at core body temperature. Today, I will talk about the regulatory mechanism of some temperature threshold for TRIM2 activation by these endogenous factors and also by TRIM2 phosphorylation by PKC. ADP levels is a well-known endogenous TRIM2 activator and appear to have a role in the regulation of temperature threshold for TRIM2 activation. This figure shows the TRIM2 activation by heat stimulation in the presence of 10 micromolar ADP levels and the physiological concentration of calcium. And temperature threshold for the current activation is analyzed by Arrhenius plot displaying logarithm of current amplitude against the reciprocal of the absolute temperature. And the friction point of the current of the trace is calculated as a temperature threshold to be around 37 degrees Celsius in this trace. The increase of ADP levels, cytosolic ADP levels from 0 to 1 micromolar reduced the temperature threshold from 43 to 37 degrees Celsius. Moreover, cytosolic calcium also has a role in the regulation of TREM2 threshold. Because TREM2 permeates calcium ion following TREM2 activation, the local calcium concentration near the activated channel pore is increased by channel activation. So in order to systemically analyze the effect of cytosolic calcium, heat evoked current was evaluated in the absence of extracellular calcium. Hereafter, I will represent this condition as a cytosolic calcium clamp. Increase of cytosolic calcium concentration from 0.1 to 100 micromolar decreased the temperature threshold from 44 degrees Celsius to 34 degrees Celsius in concentration-dependent manner. So these results indicate cytosolic ADP levels and calcium decrease temperature threshold for TREM2 activation. Recently, we have reported the regulatory mechanism of TREM2 threshold by TREM2 phosphorylation. This figure shows the results of post-hoc analysis revealing TREM2 phosphorylation by PKC. PKC activator PMA, but not PKA activator phosphorylation increased the TREM2 phosphorylation like this. And the signal increased by PMA treatment was completely abolished by protein phosphatase treatment, confirming that these signals are specifically attributed by TREM2 phosphorylation. PMA-induced TREM2 phosphorylation was persistent up to 6 hours after PMA treatment. Because PMA treatment did not affect the ADP levels induced current amplitude, I focused on the temperature threshold for TREM2 activation. As I showed in the previous slide, heat stimulation activates TREM2 with temperature threshold around 37 degrees Celsius. However, PMA treatment caused robust increase of temperature threshold to 43 degrees Celsius. Threshold elevation by PMA was observed in the presence of 3 micromolar ADP levels and 10 micromolar ADP levels. However, PMA treatment did not affect the current amplitude of heat evoked current. Because, again, TREM2 permeates calcium ion upon channel activation, calcium influx through the activated channel pore could affect the temperature threshold. Therefore, the effect of PMA was examined in the cytosolic calcium clamp condition. PMA treatment elevates TREM2 threshold even in the extracellular calcium clamp, extracellular calcium-free cytosolic calcium clamp condition. So these results exclude the possibility PMA affects the calcium permeability of TREM2 and indirectly change the temperature threshold. As I showed in the previous slide, cytosolic calcium decreased the temperature threshold for TREM2 activation in concentration-dependent manner like this. Cytosolic calcium-dependent reduction of temperature threshold is completely gone in PMM-treated condition, as shown by red traces, and PMA treatment completely abolished the effect of cytosolic calcium to decrease the temperature threshold, ranging 0.1 to 100 micromolar cytosolic calcium. These results suggest the phosphorylation counteract the effect of calcium, decreasing temperature threshold like this. Again, PMA treatment did not affect the current amplitude. RNA scanning mutagenesis of potential phosphorylation site serine residue in TREM2 identified 4 candidate residues playing a role in threshold regulation. Channel function of these mutant channels was confirmed by particular recording, following recording, excepting T689A mutant, which shows low protein expression. Even though 2 mutations affected the functional channel activity, that mutation of serine 738 mutation to alanine and its phosphomimic mutation to asparagine conserve the channel function like this. Using these functional mutation of serine 738 to A and TD mutation, and the effect of PMA was evaluated in cytosolic calcium clamp condition. TA mutant completely abolished the effect of PMA, and PMA-induced elevation of temperature threshold is almost gone in in TA mutant. For the purpose of comparison, data obtained in wild-type TREM2 channel are shown by gray traces. Gray solid line indicates that the results in PMA-treated wild-type channel showing completely different feature from TA mutant with PMA treatment shown by red bars. In contrast, TD mutant showed phenocopy the effect of PMA on wild-type channel. As seen in lower panel, TD mutant shows the slight current activation up to 100 micromolar cytosolic calcium. So higher concentration of calcium at 1 millimolar was applied to the TD mutant. Lowest current activation of TD mutant was achieved by 1 millimolar calcium, cytosolic calcium. However, the temperature threshold for TD mutants still showed higher temperature comparable to those observed in wild-type with PMA treatment. So these results indicate phosphorylation of serine 738 potentially mediates PMA treatment induced elevation of temperature threshold for TREM2 activation. This figure shows the previously reported cryo-EM structure of ADP ribose and calcium bound human TREM2. Using this structure information, the serine 738 residue is in mass TREM2 was displayed in tertiary structure of single subunit of TREM2. Serine 738 residue was predicted to be close vicinity of a calcium binding site formed by transmembrane segments S2, S3, and together with trip helix. Considering our results and this structure information, phosphorylation of threonine 738 could introduce a bulky negative charge in this site and affect the calcium binding and elevate the TREM2 threshold. Hydrogen peroxide, one of reactive oxygen species involved in the redox signal, also affects the temperature threshold for TREM2 activation. Heat-evoked TREM2 activation is dramatically enhanced by hydrogen peroxide treatment, and this figure shows the cytosolic calcium elevation accompanying temperature increase. And higher temperature, as high as 47 degrees Celsius, caused TREM2 activation, even in the absence of hydrogen peroxide. And hydrogen peroxide treatment lowered the temperature threshold towards physiological temperature. Next two slides shows the potential physiological function of TREM2 activity at body temperature. This figure shows the calcium cytosolic calcium imaging using vital mouse peritoneal macrophages. Hydrogen peroxide treatment induced calcium elevation was further enhanced by slight temperature increase from normal to febrile temperature. And macrophages have a function to eat and to internalize pathogens for phosphordefense. This macrophage function, phagocytosis, is well known to be enhanced by temperature increase. So phagocytic activity was compared between wild type and between normal to febrile temperature using zymosan, which generates the endogenous redox signal by endogenous pathway. Phagocytic activity in vital macrophages increased by temperature increase to febrile range, but TREM2 knockout cells did not show such increase. So TREM2 activity is enhanced by temperature increase from normal to febrile temperature and elevates phagocytic activity of macrophages. TREM2 is also expressed in pancreatic beta cells, having an important role to release hypoglycemic insulin. Redox signal is known to be generated by endogenous pathway accompanying glucose metabolism in beta cells and elevates insulin release. Therefore, glucose-induced insulin release was evaluated, analyzed in wild type and TREM2 knockout pancreatic beta islets to see the effect of temperature and the antioxidant N-acetylcysteine, NAC. Insulin release from wild type islets was largely inhibited by NAC, but NAC and the NAC-sensitive fraction of insulin is enhanced by temperature increase in wild type islets. TREM2 knockout islets almost abolish the effect of NAC like this. This is the last slide in my presentation, and as I introduced today, multiple factors could regulate the TREM2 activity at the core body temperature. Redox signal, cytosolic ADP levels, and the calcium ion decreases the temperature threshold to elevate the activity at body temperature. In contrast, TREM2 phosphorylation increased the temperature threshold for TREM2 activation by counteracting the effect of cytosolic calcium, decreasing temperature threshold. Because cytosolic calcium elevation accompanying TREM2 activation could cause the PKC activation, this pathway might be a regulatory negative feedback of regulation of TREM2 function. TRPM2 is expressed in cells and tissues kept at relatively constant body temperature, as shown here. TRPM2 activity at the body temperature has been gradually revealed in its involvement in physiological function, such as thermosensation in the brain and peripheral thermosensation reported from Dr. McNaughton's lab. And in order to exert adequate function of the TRPM2 channel, according to environment situations, temperature threshold for TRPM2 activation could be regulated by these endogenous factors and by currently unknown mechanisms. Finally, I'd like to my colleague and the collaborators. Thank you for your attention.
Thank you very much. Are there any questions? Yes, here. Hi. Hold on, wait a minute, please.
Thank you for this really nice presentation. I was just confused about something. You had shown that when you increase the temperature, the current density decreases. In the earlier slides, the current density decreases as you increase the temperature. So I was wondering
current density is not changed by PMA treatment.
No, not the
sorry.
One of the earlier slides. I just was wondering how it relates to the fever, if TRP becomes more activated with high temperature.
Which slide do you mean? Current density. This one?
No.
Which cells you mean?
You were talking about PMA here, right? But you had more curves. I could talk to you about it after. I could talk to you about it after.
Later.
Thank you.
Any other questions? Yes. Wait a minute, please, Peter.
It's not so much a question, more just a comment. The reason why we get a fever when we have a bacterial or a viral infection has never been explained. I searched the literature quite closely, and there are many theories about this, but the answer is certainly not known. I mean, I think the answer is in your hands here, that elevation of TRPM2 function in macrophages and neutrophils, because it's extraordinarily temperature sensitive. This enhances the activity of the innate immune system and therefore provides a strong protection against pathogens. And I think your discovery that phagocytosis in macrophages is enhanced very markedly by temperature is really, really important in answering this sort of problem, which has been around for centuries, really. What's a temperature for? So, thank you.
Yeah, one group reported the Listerine infection, the recovery against the Listerine infection. The TRPM2 knockout decreased the survival rate in the Listerine infection model.
This was discovered a number of years ago that knockout animals are markedly sensitive to infections.
So I'm going to ask one final question. So there are a lot of different PKC isoforms, and PMA hits them all, roughly. Do you have any idea what might be regulating your ion channel?
Actually, we don't focus on the subtype of PKC. And I remember the subtype of PKC has a different activation mechanism by calcium and diacylglycerol. So the cells, which type of PKC is expressed in special kind of cells?
I don't know, but there are pretty good, relatively selective blockers, isoform selective, so you can, at least sift a few out if you wanted to. look at that. Maybe other
because
people know.
we used hex cells in this
Yeah.
experiment, so
So you might be able to find out just by doing some PCR. Me with my knowledge. Any other questions? One last question here. Can you wait a minute while we get the microphone?
Which dyes did you use to measure temperature, or any other ways that you measure temperature of cells?
In patch count recording, we placed small thermometer, very close vicinity in recording, cells recording. So.
And in some images that you showed, you also had some distribution of temperature, right? So you must have used certain temperature-sensitive dyes, or?
Our collaborator has focusing on such temperature sensor to detect the heterogeneous temperature difference in the cells. But we recorded the temperature with continuous perfusion with extracellular solution of perfusion, so roughly measured the close vicinity of cells recording.
I see. Thank you.
Thank you.
Okay. Thank you very much. Excellent talk. Thank you. Thank you all the speakers.